Welcome to all the attendees to this new lecture in the context of the, what's going on? In the context of these webinars on the forefront of virology. And it is a great pleasure, really a great pleasure to welcome today Dr. Ralph Baric. Dr. Baric is a professor in the Department of Epidemiology at the University of North Carolina. He has been trained there. He had his PhD in microbiology and immunology, then a postgraduate fellowship at the University of Southern California School of Medicine, and then came back to the University of North Carolina, Chapel Hill, where he has been an assistant professor, then associate professor, and now a full and distinguished professor. Dr. Baric will present his work and the work of his group, but this is a very interesting approach, integrating really genetic, immunology, molecular biology, and biochemistry to investigate the molecular mechanisms which regulate virus replication, pathogenesis of a number of viruses, including coronaviruses. And today, I guess he will also present the new strategies they have developed to really investigate in detail the viruses, both SARS-CoV-1, SARS-CoV-2, and MERS-CoV genomes, and some very interesting results on this topic. And finally, they are also working on the investigation of the use of human monoclonal antibodies in the field of vaccines and immunotherapy. So Dr. Baric, again, we are very grateful for this lecture and we look forward to it.
Well, thank you for the introduction, Christian. It's a real pleasure to be here. And let me see if I can get my slides up. And out of the way, and then we'll be ready to go. So again, it's a real pleasure to talk to the group here. I think the development of international collaborative groups to study virus emergence, pathogenesis, and its impact on human global societies, I think is absolutely critical, especially for the future. And this is just a picture of where we work. Down below, I have a variety of collaborations with commercial groups that are investigating various small molecule inhibitors through vaccines to immunotherapeutics to treat COVID-19 pathogenesis. And I just wanted to note them beforehand. So our laboratory has basically studied emerging viruses for the last 30 years. And we do more than coronaviruses. Clearly, we're interested in coronaviruses, including MERS, SARS coronavirus 1 and SARS coronavirus 2. But we've also extensively studied noroviruses for 20 plus years, which cause about 250,000 deaths globally through diarrheal disease, mostly in our resource poor nations of the world. We also study dengue and Zika, dengue causing about 390 million cases a year. And do a smattering of research on influenza and Ebola. Much of the work is focused on mechanisms of cross-species transmission, virus evolution, and immune escape. But we're really interested in trying to develop novel technologies to improve pandemic preparedness, including predicting virus emergence potential of zoonotic strains, studying virus-host interactions, and how that regulates pathogenesis. In some work I'm not going to talk about today, we've done extensive work on studying the host susceptibility loci that regulate emerging coronavirus pathogenesis using model systems that mimic outbred human populations. This includes the collaborative cross mice, and we currently are around 60 quantitative trait loci that regulate the pathogenesis of the sarcocoronaviruses. Let me get my pointer up here. I think this is it. Yeah, there we go. And so we're also interested in understanding the underlying contributions that different host susceptibility loci contribute to the regulation not only of SARS and MERS coronavirus pathogenesis, but Ebola as well. And then finally, our program's heavily engaged in developing therapeutics and vaccines and developing platforms that allow for the identification of broad spectrum drugs, antibodies, and vaccines that protect against family-wide emerging disease outbreaks of the future. And I just want to note again that more people have died in the 20th century from infectious agents than all the wars combined. And certainly the 21st century looks like we're well on that pathway again. So if you look in the 21st century, there's been a fair number of outbreak, epidemic, and pandemic viruses, somewhere between 10 and 12, that have emerged in human populations. This includes 3 coronavirus outbreak or pandemic or epidemic strains, including SARS coronavirus of 2003, MERS coronavirus of 2012, and now SARS coronavirus 2, the cause of COVID-19 in 2019, 2020. But we've also had pandemics of alpha viruses, influenza viruses, and flaviviruses, somewhere between 2 and 4 epidemics or outbreaks of Ebola virus. And hardly anyone ever talks about the norovirus pandemic waves that have occurred, at least in years 2002, 2006, 2009, and then a fairly large wave as well in 2012. And so clearly the 21st century has seen an acceleration in the emergence of viruses that move out of zoonotic reservoirs into humans. And in the case of the human coronaviruses, there are also 3 swine coronaviruses that have emerged in the 21st century to cause huge economic losses in the poultry industry and in the swine industry. But so clearly, SARS coronavirus 2 has outpaced all of these. It's currently caused about 2.5 million deaths worldwide. This is sort of captures the death toll from emerging viruses, a representative group over time, showing that SARS and the MERS causing minor epidemics here of about less than 1,000 cases of death, and how COVID-19 has eclipsed many other major pandemics that have occurred throughout human history. We currently, again, have about over 500,000 deaths in the United States, and probably the best case scenario argues that we've got at least 100,000 more deaths before we see the back end of this pandemic and herd immunity is achieved. And of course, that is dependent on how the virus evolves in response to countermeasures that humanity throws against it. Where exactly will COVID end up as a global pandemic? We don't really know. Probably somewhere between the Antonian plague and the Third World Plague. But I don't believe that it will eclipse or pass things like HIV, AIDS, or smallpox or Spanish flu pandemics, because for the most part of medical countermeasure capabilities. So today's talk is going to focus on SARS coronavirus strains and variants and their anagenic relationships between them. I'll talk a little bit about animal models, and then just briefly talk about some countermeasure applications that we've used in the animal models to try to bring products into human use. And so the first variant that our group became interested in was described by Betty Korber in a Cell paper around 2020, which was the D614G variant. It was noted that prior to early February, middle February, this variant was very rare in populations, and then emerging somewhere, most likely in Southern Europe, although that's debatable. This G614 variant became the predominant strain worldwide, and this is a mutation in the spike glycoprotein. And the early data suggested that it was associated with higher virus titers in the upper respiratory tract, but it was not associated with increased disease severity. So we decided to use reverse genetics that we developed in our lab to ask questions about the role of that mutation in replication and pathogenesis. And using a molecular clone that Jacob Hu in the laboratory developed, we basically made isogenic viruses that either had the D or the G variant at this position, noting that we destroyed a restriction site here in the wild type virus to produce this G variant, which is used as a measure for some competition studies we did later on. We also built these viruses in the context of nanoluciferase so that we could do a variety of other assays, one of which was just a basic infectivity assay where you infect cells and shortly thereafter look at the amount of nanoluciferase expressed to get an idea of how infectious the virus might be in different cell types. And in every continuous cell type that we looked at, you could see that the virus was more infectious than the original ancestral strain that came out of Wuhan. Jacob, working with Marsico Lung Institute at UNC, had done a very careful study where not only did he develop the molecular clone, but he worked to characterize the gradient of cells in the human upper and lower respiratory tract that were susceptible to SARS coronavirus 2 infection. And carefully, he and others measured the amount of human ACE2 from the nose to the most distal regions of the lung and the alveoli, showing that human ACE2 is expressed highest in the nasal epithelium, lower in AT2 and AT1 cells, and little if any growth was noted in lung fibroblasts or microvascular endothelial cells. And he used the GFP virus here, which, is, you can see, affects primarily ciliated cells in the airways. Using this model system, he has basically compared and contrast the ability of the D and the G variant to replicate in multiple codes from different individuals from the nasal airway epithelium, the large airway epithelium, and the small airway epithelium of the lung. And his data clearly showed that in the nose, the G variant replicated to significantly higher titers and much faster than the D variant. It also replicated to significantly higher titers in the large airway epithelium, but not so much deep in the lung, supporting some of the early work that suggested that the G variant replicated to higher titers in the upper, but not necessarily the lower respiratory tract. He used competition studies, classic competition studies, where you infect cultures with equal amounts of the D and the G variant. And then every 3 days passaged virus onto fresh cultures to look at the fate of the two progenitor viruses and to see which one was more fit. And doing these competition studies at a 1-to-1 or at a 10-to-1 ratio, we'll just focus down here at 10-to-1 because it's clearer. You can see that the G variant rapidly outcompetes the D variant in every assay. And so this demonstrates at least one of the reasons why the G variant would have been capable of outcompeting the D variant and becoming the most prevalent strain globally. We'd also created nanoluciferase viruses so we could look at the impact of the mutation on virus neutralization. We have developed both optimized, qualified, and nearly nearly validated assays, neutralization assays through the Food and Drug Administration for use in evaluating immune responses in human volunteers. And so hopefully we will have a validated assay by the FDA for some studies with human vaccine samples shortly. But anyway, we use these assays to look at neutralization responses of the D and the G variants using contemporary human sera, mostly from individuals that were infected with the G variant, but some were infected with D. And you can see that the D variant is certainly more resistant to neutralization as compared to the G variant. And this is significant. This is both at the ID50 and at the ID80 levels. And in fact, the G variant is about 2-fold more sensitive to neutralization than the original ancestral strain that came out of Wuhan. And this is just some Spearman correlations showing how well the assays at ID50 and ID80 work. We worked with Yoshi Kaoka to look at the ability of the isogenic viruses encoding different residues in spike on pathogenesis in the hamsters. There was no difference in titer in the lung of these animals or in the nasal turbinates, and only a slight increase in weight loss following infection. When you look at the lung pathology of these animals, it really was not significantly different. So we think it's, if anything, it has a minor impact on overall pathogenesis in animals. More importantly, we set up transmission studies to look at the ability of a donor-infected animal in this one cage to transmit via airborne transmission to a recipient animal in a second cage with an airspace in between. And when you infect these animals, you can see they all get infected. This is with a wild-type ancestral strain. It clears by day 8. The titers are not significantly different with the G variant through day 8 and also clearing by day 8. But when you look at transmission events, you can see that none of the ancestral viruses had been transmitted to the recipient animals by day 2 post-infection, but all had become infected by day 4. In contrast, 5 of the 8 or 8 of the hamsters became infected by day 2 with the G variant, really showing strong evidence of enhanced transmissibility. We've also done competition studies in the hamsters, and the G form outcompetes the D form as well in vivo. Now, we have provided Yoshi a large panel of variants and strains of SARS coronaviruses to do similar studies, and hopefully that data will be rolling out soon. The G variant was certainly important because it really serves, for the most part, as the foundation for all the existing new variants that have been described in the literature, including variants from the mink cluster 5 variant, which is not on here, the UK variants, the South African variants, and the Brazilian variants, as well as derivatives throughout the United States that have clusters, slightly different clusters of these mutations. Detailed studies on transmission and virulence argue that the UK strain is more transmissible. It's probably 30% more virulent. It's about twofold more resistant to neutralization. The South African variant had the highest increased resistance to sera, either from convalescent patients or from vaccinees, of about a 6 to 10-fold reduction. And the Brazilian variant has about a 4-fold reduction in neutralization titers as compared to the other variants. Our own lab is working on both the UK, South African, and mink cluster 5 variants in terms of pathogenesis and replication in multiple airways using the systems that I just described. But since so many groups have already used lentiviruses or VSV to characterize sort of the properties in terms of neutralization escape, I'm going to focus on a larger picture to try to identify what the what sort of resistance might be possible with SARS coronavirus 2 as it evolves during the expanding pandemic. And to do that, we wanted to ask what the effect of the anagenic variation is across different SARS coronavirus strains, which fall into 3 clades based on my nomenclature. Clade 1 strains and timing of discovery. Clade 1 strains include the original 2003 strain, as well as some bat strains that are shown here with arrows. The Clade 2 strain include bat strains, most of which do not replicate in primary human cells. And then we have Clade 3, which includes SARS coronavirus 2. We have resurrected the pangolin virus as well, and we have nanoluc viruses, nanoluciferase expressing viruses. Of all of these strains, as well as a variety of group 1b and group 2c nanoluc reporter viruses. And so we wanted to use these to look at the effect of maximal anagenic variation on neutralization using both monoclonal and polyclonal sera. And the first study that I'll present here was in collaboration with Laura Walker, Jason McClellan, and others, where Laura's group had initially isolated a panel of antibodies that would bind to the receptor binding domain of SARS-2 and neutralize virus infectivity, but we were interested in using a directed evolution approach to engineer human monoclonal antibodies that could neutralize the variants, the high-risk pre-epidemic variants that existed within bat populations or had caused previous epidemics. And so using directed evolution and sequences from high-risk strains that we'd identified, we isolated a series of new antibodies. The one that I'm going to focus on is ADG2. It's being compared to antibodies from Regeneron or Eli Lilly or other groups. And if you just look at the SARS coronavirus 2, ADD2 is in red. You can see it's a potent neutralizing antibody against SARS coronavirus 2, as are many of these other commercially available or reported antibodies that are on this list. When you compare the neutralization profiles with the 2003 SARS coronavirus strain, you can see that the ADD2 antibody is still extremely potent, if not more potent, against SARS-2, while most of these antibodies have lost neutralization potency against the original 2003 strain. When you start looking at high-risk pre-epidemic bat coronaviruses from clade 1 that can replicate in human cells, can use the human ACE2 receptor and cause disease in human ACE2 mouse models of disease, you can see that the ADcd2 antibody is a potent neutralizing antibody against both of these bat viruses, while many of these other antibodies like 309, which still has some potency across the panel, has lost over 100-fold of its potency. Jason has solved the structure of this antibody. It binds to a portion of the receptor binding domain of the spike glycoprotein, and it covers a portion of the ACE2 binding site. And so its mechanism of action is to prevent interaction with the human ACE2 receptor. Now, this represents the first broad-based human monoclonal antibody against the SARS coronaviruses, and it neutralizes all the strains, at least that we have within our portfolio. And hopefully that could be used to not only treat patients in the COVID-19 pandemic to demonstrate its efficacy, but also to have it on the shelf for future pandemics. Now, we'd like to show that that antibody actually works in vivo. So Sarah Leist and Kenny Dinnan in my lab had created a lethal mouse model of human disease. I'm just summarizing this. This was published in Cell this fall sometime. Basically, this was done through genetic engineering of the receptor binding domain to grab onto the ACE2 receptor, the mouse ACE2 receptor, followed by serial passage in vivo. And this model causes 70 to 30% weight loss and a 90% mortality in old animals, a reduced weight loss and mortality in young animals. So it captures the human age-related disease gradient and replicates to very high titers in the lung in a biphasic disease model. So titers are high early, it drops off, and then disease occurs late. So it sort of captures many of the features that are seen as a biphasic disease process that are seen with SARS coronavirus 2 in humans. Importantly, I'll just show a couple of pictures here. The virus targets the olfactory epithelium in the nose, but not neurons. Neurons are shown in green here, overlaid. It looks like it's the sustenance cells that sort of wrap around the olfactory neurons and provide support functions. And so we think loss of olfactation may be associated with the destruction of the olfactory sustenance cells rather than through direct infection of the neurons. But we're still working on validating that. In the deeper lung, in the alveoli, the virus infects AT2 cells. This is LAMP3, it's a stain for AT2 cells. You can see you have co-localization of LAMP3 and viral antigens in red. Interestingly, when you look at surface expression markers on AT2 cells following SARS coronavirus 2 MA10 infection in the lung, the surface marker expression rapidly disappears, including LAMP3, but also surfactants C and B. They're shown here. C is in green here for surfactant, B staining is green here. Red is virus infection. You can see all the green disappears, mostly from the parenchyma of the cell, although the AT1 cells are saved. And so we think this results in rapid surfactant C and B loss. You can knock out, I think it's surfactant C in the animal, and a mouse will survive just fine, but if you knock out surfactant B, you have a dead mouse. The alveoli can't open up as you breathe in to bring air into the deep lungs. So we think surfactant deficiency may contribute to death. And to summarize a lot of the pathologic features of this model, it attacks club cells in the terminal bronchi. It can infect ciliated airway epithelial cells, olfactory airway epithelium, and AT2 cells are the target tissues in the lung. It results in acute lung injury to ARDS, depending on the age of the animal. There's increased cytokine storm and surfactant deficiency, really all contribute to mortality. And we're currently trying to identify the effect of infection on long-term chronic disease sequelae. Now, so having created a mouse-adapted model for SARS coronavirus 2, we went back to the Adcd2 antibody and asked whether it could protect against lethal SARS coronavirus 1 or MA15 infection, or so SARS coronavirus 2 infection, shown here with the MA10 virus. All of the MA15 infected age animals were dead by day 3. That were treated with irrelevant antibodies, but the Adcd2 antibody provided complete protection, not only against the original 2003 strain, but also against this MA10 strain. Under therapeutic conditions, it also provided protection after 24 hours, although the animal still had virus titer and replication, and I'll run through this pretty quickly. Respiratory function is measured as airway occlusion. From debris in the airway. It's a resistance marker, so this is normal down here. And this is an animal that's been infected with virus. You can see it's having trouble breathing. Same with the MA10 virus, but they're completely protected with Adcd2. And in this therapeutic conditions, you have partial protection. You have complete protection against lung congestion under prophylactic and reduced and protection therapeutically. And you have protection against virus replication, both prophylactically and significant reductions, especially at day 4 therapeutically. So the antibody also works in vivo. Now, to get a broader picture of the anagenic diversity of the SARS-related viruses, and to get an idea of what we might expect with SARS-2 evolution over time, these are a list of all the high-risk sarcoronaviruses that are capable of using human receptors for entry, and which we have available in the lab, most of which with nanoluciferase. And so we could take, and if you look at the Urbani spike as compared to the 2002 spike, 2002 strain, if they're about 22% different, SHC014 and WIV1 are about 8 to 10% different. But notably, if you compare these two, they're over 25% different, and this is a little bit less. And so these bat strains have a tremendous amount of anagenic variation, increasing up to about 35%. So this is some work that we just put up on Baric in collaboration with Bart Haynes's group that looks in primates vaccinated with various vaccines and looking at their ability of the SARS-2 nanoloop vaccines to protect primates or to neutralize virus from different sarcoronavirus strains. And so we're looking here at neutralization titers of around 10,000 to 15,000 against SARS-2 nanoloop virus. If you compare it to SARS coronavirus nanoloop down here, you've got about a 20 to 30-fold reduction, depending on the vaccine. If you compare this with the WIV bat strain, it's about a 10 to 20-fold reduction across these different vaccine formulations. And the SHC14 is about a 200 to 300-fold reduction. We've done identical studies with mRNA vaccinated mice and shown, starting again at about a neutralizing titer of 1 to 10,000, the SHC014 viruses are about 500-fold reduced in terms of their resistance to neutralization. SARS is still in the 20 to 30-fold range, and WIV1 is in the 20 to 50-fold range. So in essence, this probably represents the upper range of limit in terms of potential escape that coronaviruses may be able to achieve as a function of time. And so we're currently obviously doing similar studies with the variant strains that have emerged, which we feel are in the early stages of evolution, and that much more potent resistant strains will emerge in the near future. In terms of the vaccine performance, in terms of the variants, I just wanted to show this data here that in the Moderna-based vaccines in our hands, we get titers of around 1 to 5,000. The South African variant, which causes a 6 to 10-fold reduction, would reduce that to maybe 1 to 500, which would still be fully protective. I think Moderna and Pfizer are both clearly will most likely not be impacted by the current mRNA-based vaccines because most people mount really high neutralizing titers in the top quartile of natural infection. Novavax is about 89% effective or 90% effective. But in South Africa, the South African variant broke through reduced the efficacy to about 60%, and most of those were the variant that broke through and killed. And Janssen vaccines, I don't think have been looked at yet, or if they have, I didn't have time to find and read the paper this week. So that's sort of the current status. We've also used the model to evaluate drugs. In this case, it's against remdesivir, giving BID subcutaneously under both prophylactic and therapeutic conditions, and measuring body weight, pulmonary function, lung, virus titer, and pathology. This was done by David and Alex. And in under prophylactic conditions, you can, shown here in the orange bar, you can see that remdesivir is completely protective in the SARS coronavirus 2 model. It's less effective 12 hours after infection, less effective, but still effective at 24 hours, and it loses effectivity by 48 hours. And that occurs not only with weight loss, but also reductions in virus titer are actually still significant at 48 hours, although the animal has still lost tremendous amounts of weight. And this is a lung congestion score. So we see this phenotype in our mouse model regardless of what direct-acting antiviral we use. It can be an antibody, it can be a therapeutic. All of them have a short therapeutic window. And that really reflects the kinetics of the disease in both humans and mice. In the case of humans, about 4 days prior to infection, when you first become infected, before you're asymptomatic, virus titers peak early and then drop late in a human. In the mouse, it's much more acute. Disease occurs after acute high titer virus replication has occurred. And so it's a biphasic disease model where virus titers go up fast and then come down, and then disease comes up late. Direct-acting virals, therefore, have a narrow therapeutic window. Remdesivir, which is given by IV following hospitalization, really is at the end of that therapeutic window, and we need to extend it to earlier times, either by the development of oral direct-acting antivirals like EIDD2801, which is currently in phase 2 testing that we did all the preclinical work, or in combination with anti-inflammatories, which we're studying as well. So this just gives a quick summary of what we've done and what we've worked on in terms of the preclinical development of remdesivir, 2801, pegylated interferon lambda, and a variety of therapeutic antibodies from different companies. So with that, thank you for your attention. I know I went a couple minutes past 30 minutes, but hopefully that's okay. And again, the key people in my lab are highlighted here. I've mentioned them throughout the talk. We have great collaborations with Yoshikaoka and University of Wisconsin, Madison. The Marsico Lung Institute's been a key player for us. I've worked with Mark Dennison's lab for years. We have collaborations with Duke, Pittsburgh, and a variety of companies. So again, thank you for your attention. I'll be glad to take questions.
Thank you so much, Ralph. That was really a great talk and highly informative. So thank you really so much. Now I will hand over to Shin-Hye Kim, who is the scientific director of the GVN, to handle the questions and answers. Shin-Hye?
Yes. Thank you, Dr. Baric. It was a really great presentation, and thank you for your good work. And the first question came from Dr. Gallo, and the super talk route question is, in view of the lack of a neurological infection yet, several neurological symptoms in some, do you think that these are all due to the related inflammatory cytokine, like much other pathology?
No, I think that's a great question. I think the data is mixed, certainly in humans, about whether the neurons are infected. There's no question that the K18 model, which is a human ACE2 transgenic model, the virus can spread to the central nervous system and cause an encephalitis. It also infects olfactory succenticular cells and olfactory neurons to cause a loss in olfaction in that model. I think the big unknowns are whether the cytokine storm or whether particular cytokines may elevate ACE2 expression in the central nervous system to allow for virus to spread into the CNS in some individuals. But I would probably argue that the major CNS symptoms are associated with poor oxygenation of the central nervous system that's associated with ARDS, clinical disease symptoms. And so the central nervous system becomes hypoxia. I would also argue that probably the major cause of additional neurologic complications would be clotting, coagulopathy-related complications that occur during SARS coronavirus 2 infection. And the combination of those factors, I think, can lead to these sort of long-term neurologic sequelae.
The second question is that, could the Adcd2 antibody protect against the infection by SARS coronavirus of other susceptible hosts with slightly different ACE2 receptors like MINC?
That's a great question. In fact, any therapeutic antibody that targets the receptor binding domain has the potential to select for mutations in the receptor binding domain that would still allow use of the human ACE2 receptor because you're doing it in a human. But subtle changes in those ACE2 binding residues without question change the host range potential of SARS coronavirus 2. And there's good examples of that already in the literature. So the emergence of the 501 mutation in the receptor binding domain confers the ability of SARS coronavirus 2 strains to replicate in the mouse. So things like the pangolin strain, for example, which has the 501 mutation, can replicate in the mouse. Some of these variants that have the 501 mutation replicate just fine in the mouse. They don't produce serious disease, but they can replicate in the mouse. And so every time we see a change associated in a variant with a residue, a contact interface residue that binds ACE2 receptors, we could have subtle changes in the animal tropisms of these strains leading to new reservoir species. And clearly the MINC cluster 5 variant is sort of the poster child for that hypothesis, in that the strain emerged in Denmark, it infected minks on mink farms, and then transmitted back to humans who transmitted back to minks. So in essence, a whole new ecologic reservoir was being created to allow for maintenance, evolution, and then transmission back to human populations. And so we really need to pay attention to this. And I think the other factor we really need to pay attention to is that SARS coronavirus 2, which was localized in China and Southeast Asia, to the best of our knowledge, certainly that's where all the bat-related strains have been identified to date, is now seeing mammals and bats that it has never come in contact with across the globe. And so there could be bat species that have been colonized with this virus. The virus could be undergoing recombination events with other group 2B or group 2 coronaviruses in bat reservoirs, only to come out and to inflict harm on human populations in the future. So this is a huge threat, and it's a great question. I have not actually looked. We have not, we are looking, but we have actually not finished looking at what sort of escape mutants emerge with the Adcd2 antibody. Having worked with a lot of antibodies with both SARS-1, MERS, and SARS-2, I'm 100% confident I can make an escape mutant if given enough time and energy.
This is a sequential question. Does the Adcd2 drive the selection of a spike variant?
Oh, yes. I sort of answered that because I read the question. We're working on that. I've been, so far there was one antibody. To give you an example, in my opinion, you can make escape mutants from almost any antibody. And it's just a question of the amount of time required to do it. So the worst case, there was an antibody that Antonio Lansavecchi identified called 227, which SARS coronavirus 1 was completely susceptible to, and we could never isolate escape mutants to it. And George Giorgio also made an antibody that was incredibly potent through antibody engineering-based approaches against SARS-1, both of which we were never able to isolate, had very, very difficult time isolating escape mutants. The way to do it is to reduce the amount of antibody and take your time, slowly increasing the amount of antibody over 20 passages. You will get novel resistant mutants. Usually two or three amino acids will be required to change. The other way to do it, which is easier, is to select for resistance in different backbones. So for example, the Adcd2 antibody, if it's really difficult to make a SARS-2 escape mutant, my gut feeling was that if we would go into any of the bat strains or the 2003 strain, the different backbone residues would make escape much easier. So yes, the answer is we are looking at making escape mutants to that virus, and I believe we will find them.
Have you looked or planning to at the effect of FC on prophylactic therapeutic potential of monoclonal antibodies?
There have been a couple of studies that we participated in looking at FC receptor contributions to protective immunity, one done with Galit Alter, another with a group out of the Rockefeller. And in this particular case, the Adcd2 antibody, Lara's group did a fair amount of extensive work looking at the FC effector functions of the Adcd2 antibody. And you can take a look at that data in the paper. It's good.
The GeoLethal mouse model show evidence of coagulopathy like D-dimers.
There's no question we see evidence of microthrombi. We also see that with the original SARS coronavirus 2003 strain. We actually published in 2013, I believe, that one of the major contributors to SARS coronavirus pathogenesis was a dysregulated coagulopathy. And that included a large amount of systems biology work as well as targeted knockouts in the urokinase pathway that regulated disease severity. We have used those same knockouts with SARS coronavirus 2 that certainly supports the argument that there's a dysregulated coagulopathy. We have not measured D-dimers, but Lisa Grlinsky, who's a assistant professor at UNC, is doing a tremendous amount of collaborative work with the hematology group at UNC to look at all of those metrics, as well as testing viral pathogenesis with the MA10 model in various knockouts in the key pathways there.
What about additional receptors or entry cofactors such as neuropilin, CD147? Any evidence for in vivo relevance?
I have not done the experiment with SARS coronavirus 2, but we did the experiment with SARS coronavirus 1 some time ago. And an ACE2 knockout mouse does not become infected with SARS coronavirus 2 MA15. I'm sorry, SARS coronavirus 1 MA15. So in our opinion, in the mouse model, at least with the original strain, the ACE2 molecule is the only receptor that plays a critical role in mediating infection. Now, that doesn't rule out the possibility that cofactors can enhance the ability to bind to the surface of cells and come in contact with that receptor, but I think ACE2 is the predominant receptor, at least in the lung.
Did you confirm by reverse genetics whether only spike mutations are sufficient to obtain a mouse-adapted virus?
Yes. We published a paper in that in Nature sometime this fall.
You mentioned the high level of ACE2 receptors in the nasal passage. Would an anti-ACE2 antibodies be a good protective modalities?
Hey, Ray, nice to hear from you. Thanks for the question. I think that's a really interesting question. For most coronaviruses, antibodies against the receptor rapidly select for antibody-resistant variants. But there's no reason why an antibody to the ACE2 receptor couldn't mitigate or protect the nasal airway epithelium from virus infection. I actually don't know if anyone's actually testing that, either in humans or in animal models of human disease. It would be a good question to ask. I know that different groups have tried different nasal sprays, including detergents and things like that, to try to flush out virus out of the nasal airway epithelium. In our hands, when we do things like the ivory soap experiment, using primary cells, you can inactivate virus really quickly, but within a few hours, the titers are all back. So it's very transient. So that would be a couple of questions you'd have to ask in terms of how long would anti-ACE2 antibodies that bind to or engage the receptor binding domain, how long would they protect via an intranasal instillation? And so that, it's something that probably should be looked at.
Do you expect an optimal virus receptor affinity for maximal transmissibility, or is a higher affinity always be the evolutionary endpoint of the virus?
Yeah, that's a great question. I actually believe high affinity, extremely high. Let me say it this way: virus RBD ACE2 interactions, I believe, is a bell-shaped curve in terms of fitness and infectivity. And the reason I say that is some experiments we did a long time ago and published in the 90s, where if you overexpress receptor, you can infect cells extremely easily with virus. But the progeny that come out have the spike stripped off the surface of the virion. So really high levels of ACE2 or high affinity virus receptor interactions can prevent the free release of virus from cells. It can leave the virus attached to the surface of the cell that it just infected, or it could potentially strip off the receptor. In addition, binding initiates a series of conformational intermediate phases that are critical for spike reorganization and entry. And I think personally that extremely high affinity interactions are deleterious. And so the virus has to find a good balance of high affinity, but not so high affinity that it can't get off of that receptor. So that's actually a dangerous experiment to do, because you have to engineer viruses with extremely high affinity ACE2 binding receptors. And if my hypothesis is wrong, I could have made a more dangerous virus. So I hope anyone out there with a molecular clone isn't going to do that experiment.
What is the best antigen for universal vaccines? Thank you for your presentation.
I think it has to be spike. It's the major, so there's two models. I take that back. There's two models. One is a really good T-cell based vaccine has the potential to provide protection from lethal disease as long as it can be activated before day 6 or 7 post-infection, and given the time course between initial exposure and severe disease, a T-cell based vaccine may provide protection from lethal disease, but I think the patients will still get very, very sick. I think antibodies are the key to prevent serious disease. And so because of that, the spike is the major site for neutralizing antibodies. There were earlier studies done in 2003, 2004 that argued that the other surface glycoproteins on the virion did not provide neutralization or mediate neutralization of the virus particle. I don't know if that's been done yet with SARS-2. So I think a SARS-2 spike-based vaccine is probably the key. But there's nothing wrong with having a good universal T-cell response either.
Do you really think we can achieve herd immunity before variant emerge to reinfect animals that had SARS resistance to recurrent infections?
Oh, so there's clear evidence of SARS-2 reinfecting individuals on planet Earth. And when you look at the neutralization titers in individuals, they correlate pretty tightly with the severity of disease. So asymptomatics typically have low titer in the range of anywhere from 100 to 500 antibody titers, neutralization titers. A variant with 10-fold reduction of that puts the person certainly in the realm of susceptibility. When you also take into account that the durability of the immune response in the upper airways and the nose is much more limited than it is deeper in the lung, that would also combine to allow for virus reinfection in the nose. In other words, a common cold type illness that would then boost your immunity. So I think that will be actually quite common as these new variants emerge in populations. I think that was the question, right?
Yeah.
Okay.
And some have suggested that the vaccine will lower the number of infected so well that the virus will disappear.
No, I don't think they know what they're talking about. And the reason I say that, with a lot of hubris, by the way, is that there have been 4 contemporary human coronaviruses that have emerged from animal populations, all of which have colonized humanity, and all of which cycle on 3 to 5 year intervals to cause epidemic waves in human populations. We don't know how they maintain themselves and how they evolve in human populations. The studies, many grants submitted to NIH by the coronavirus community on that very question were rejected because common cold viruses weren't interesting. So we don't really know the full basis for how the contemporary human coronaviruses maintain themselves in the population. What little is being looked at suggests that they're evolving in an influenza A type pattern in response to human herd immunity, and that reinfections can occur within 3 to 6 years following exposure to that strain. And those strains have anagenic variation within them. Again, because of the compartmentalization of the immune response in the respiratory tract and the different rates of waning immunity in those 2 compartments, what happens is that the nose becomes vulnerable first and you end up with a common cold phenotype. So I think we're going to end up with SARS-2 being a common cold virus, infecting primarily young children in the first 5 years of life, and repeat infections in individuals until they're much older and begin to immunosenesce and become vulnerable again in outbreaks in retirement communities, elderly assisted living communities, in individuals above 80 years of age have been associated with 10% mortality rates of the contemporary coronavirus. So I think that's what's going to happen. Obviously, what I don't know is whether there are emerging coronaviruses that occurred in the past that spread globally and then went extinct, in which case, then I'm completely wrong.
Is there any interest commercially to immunize any SARS-CoV-2 susceptible farm animals, such as live in the dense population and near to humans?
There's certainly discussion about doing that in mink populations. But I think it underscores a broader question, which is we really need to do surveys of wildlife to understand exactly what this virus is doing within those populations. And that may tell us whether we should begin to consider vaccinating important livestock and or other potential zoonotic reservoirs that could lead to reintroductions of recombined and anagenically distinct SARS-2-like strains of the future. I think that's a real possibility that we need to be alert to.
There are many known missense variants of human ACE2 in the population. Could any of these be responsible for protection of some humans over others to susceptibility to infection with the SARS-CoV-2?
I missed the very beginning of that question. Can you re-ask that question?
There are many known missense variants of human ACE2 in the population?
You mean ACE2 polymorphic allele variants?
Receptors, ACT receptors. The difference of ACE2 receptors in the human to human, different host.
In terms of human polymorphisms, I'm aware of one paper that suggested minor phenotypes on the ability of the virus to infect reporter ACE2 expressed and overexpressed in cells, which is quite different than what happens in humans. I don't know of, and it could be that I just missed the literature, it's easy to do, but I don't know of any population of allele variants in ACE2 that have been proven to be resistant to infection with SARS-2. If I see Matt Freeman's on there, Matt will tell me in a second if I'm wrong. Is that right, Matt?
So we are almost at time, and so Dr. Brechot, do you want to continue or you want to stop?
No, we can take, I see that, well, first, there are very many questions, Ralph, which really just shows the great interest to your talk. We are not surprised. We can take maybe, I see 4 questions, okay?
Right, so we'll put
4 questions, okay?
So
Sounds good.
Yeah, we'll finish all the questions. Yes, but targeting a receptor might be disaster inducing, right?
So it's possible. So the virus is going to have to figure out a way around the antibody block to ACE2 binding. And they usually do that. If it's anything like the other coronaviruses, they'll do it by changing affinity for the receptor and also making some changes in S2 that probably change pre-conformational states that are more in a ready prefusion state. So you could also get viruses that potentially are evolve more towards a prefusion state that could be much more infectious. They may be less stable in the environment. I'm trying to remember the old literature of the antibody receptor escape mutants. I can't recall. Susan Weiss did a lot of this work as well as Tom Gallagher, and I can't recall whether those variants were more or less pathogenic. That's a really good question.
The loss of a surfactant after infection in mice is interesting since the virus infects the ciliated cells, not the major mucin, and the surfactant producing cells. Do you think that this is a repair response or a differentiation of the goblet cells for repairs of the lung damage caused by the virus?
Yeah, that's a great question, Matt. We think there's a whole bunch of questions in there. Number one, the AT2 cells are hit hard in the mouse model. The first question is, how does the virus reestablish the AT2 cell lineage following virus infection after many cells have died? That's a question that we're actively engaged in. Goblet cells are strange. They express a lot of ACE2 receptor. They have TMPRSS2, but they don't seem to get infected. Why is that? That's a really good question as well. Club cells are heavily targeted in the terminal bronchi, which can differentiate either to repair the more distal airway epithelium or the airway epithelium. I'm sorry, the more distal alveolar epithelium or the more airway epithelium. And so what sort of lineages are engaged in repair of the lung following infection of the mouse model is a major area that we're interested in and that we've been studying for quite some time with Marsico Lung Institute, who have a variety of novel mice and cell line types that allow for lineage tracing and those kind of questions. So I think it's a great question. I also suggest, it seems like our data suggests, and we know AT2 cells are heavily targeted in humans, that surfactant replacement therapy may offer a potential therapy for SARS coronavirus 2 patients in the clinic, but from what I understand, it has not been attempted as of yet, and maybe we should start thinking about that.
This will be the last question. Is there any evidence that people who were infected with the SARS-CoV-1 are protected or not from SARS-CoV-2 infections?
So the biggest population of SARS coronavirus 1 infected patients were in China. So that would be the best cohort. I seem to recall some statements made by Chinese researchers about susceptibility of SARS-1 patients to SARS-2. And I can't recall whether the answer was yes or no.
And we have another additional question. What do you think about humanized mouse models?
Oh, sure. I probably should have put a couple slides in there. That we've done some work with Victor Garcia at UNC, who has humanized lung-only mouse models. And in those models, all of our SARS-related coronaviruses that we consider pre-epidemic replicate efficiently in those lung-only models. And you can reconstitute the human immune. I guess the real strength of that model is that it's sort of the mouse immune system's been disabled, and you can reconstitute those mice with various human cell immune components to look at the role of the human immune system in clearing infection in human lung tissue. And so that's a real strength of that model. So I'm very, very bullish on that model for those types of virus-immune interaction studies. And we're continuing to do those studies with Victor as I speak.
That's it. Thank you so much, Dr. Baric.
Yeah, thank you very much.
Okay, a pleasure talking with you all.